Thanks very much for the introduction. My background is that I started my career in academic medicine and spent the last 20 years as both a venture investor and an operating executive in startup companies and healthcare. And to me, there are two things that get you excited about a startup. One is having a technology that's truly revolutionary, and the second is having that technology actually be applicable to a common disease state. And that's what we bring together here at Alucent. I'm here to talk to you today about the Alucent NVS vessel restoration system with photoactivated linking based on a technology called natural vascular scaffolding. And to put it very simply, the idea is that we're able to stent open a vessel without the need for a stent. The condition that we're looking to treat initially is peripheral artery disease. This is a very common condition worldwide, afflicting over 200 million patients, over 9 million in the US. And the risk factors for it and the prevalence of it are very similar to coronary artery disease, but it has a very significant unmet need. The unmet need is that for the last 50 plus years, we've been treating this disease largely with angioplasty balloons and stenting. But if you talk to real world physicians out there, as opposed to the lesions that you see in some clinical trials, they'll tell you that about half the time they fail and the patient has to come back and be retreated. And this is a particularly problem when you're talking about long lesions, lesions that are heavily calcified, or when the initial balloon therapy is accompanied by dissection, which is a common complication. And there's too many folks that end up like this poor chap here, having lost a limb and been failures. That large number of patients, 200 million globally, of course, translates also into a very large market, about $3.5 billion, growing at mid to high single digits. As you can see from the slide here, about 2/3 of that is in lower extremity peripheral artery disease, the narrowing of the arteries of the leg, and that's our initial market focus. However, it'd be remiss of me as the CEO of a company with an exciting technology not to tell you first about some of the other potential applications that we have. I'm going to spend the rest of the time talking about peripheral artery disease, but you can see here some of the other vascular conditions that we can address. One in particular that's very exciting to me is the last one on the list, which is the prevention of AAA progression, abdominal aortic aneurysm. This is an area where there is no current therapy until the end stage. And based on the phenomenon that we frequently see in diabetics, whereby their aneurysms do not grow as quickly as non-diabetics, that is thought to be due to high glucose levels leading to the linking of the structural proteins in the vessel wall, thereby stabilizing the aneurysm. Our technology is very similar, of course, without the high blood sugars, and we've actually shown in animal models, standard animal models of AAA, that we can dramatically reduce the rate of growth of aneurysm using NVS technology. Okay, well, back to PAD. As I mentioned before, we've had over 50 years of therapy with balloons and stents dating back to 1964, and the very first angioplasty, which was actually done in a peripheral vessel. The more famous example was in not sure what happened there. Anyway, the more famous example was in 1977 with Andreas Grunzig, who performed the very first coronary angioplasty. And then following on that, the creation of stents. However, all of these therapies have had really significant problems. Balloons, although they open the lesion in the vessel, result in acute recoil, they result in thrombosis, they can result in dissection and restenosis. Stents can help address some of the initial recoil problems, but still have in-stent restenosis and now stent fractures and other complications of implanted therapy. So as a result, we are ready and willing and need a new treatment paradigm in this space. And of course, you know what I'm going to tell you next. I think that new treatment paradigm is NVS therapy. How are we doing on the slides here? No? Okay. Well, I just told you all about this. Next, I'm going to tell you about what is NVS. As I mentioned initially, it's stenting without a stent. It's a fully integrated therapy that's comprised of 3 components. The first component is a photoactivatable small molecule. The second component is a light fiber that emits light at the proper wavelength to activate that small molecule. And the last is an angioplasty balloon. And what we're actually doing is to use the small molecule when activated as a catalyst, which creates links between certain amino acid moieties in the collagen, the native collagen and native elastin in the vessel, thereby creating a natural vascular scaffolding effect out of the native proteins in the tissue. We know that in the course of plain angioplasty, while we open the lesion, we actually destroy the natural structure of the collagen in the vessel, which creates a floppy vessel. And this therapy actually causes those proteins to relink and reform their normal structure. The product that we would actually like to bring to market is this. You can see on the left-hand side of the screen a fully integrated, very simple to use product. Looks very similar to other cath lab products. It's compatible with standard guide wires and standard access systems, and it combines all three components. The right side of the screen shows you a close-up of the angioplasty balloon. That angioplasty balloon is coated with our drug, which has a yellow-orange coloring to it. And the bottom picture on the right is the integrated light fiber illuminated within the device. This animation should give you a good idea of how the product will work in practice. We're going to focus in on a cartoon representation of a lesion in the superficial femoral artery. And what you're going to see there is that we're going to cross the lesion with a standard guide wire as the initial step. Then we introduce our device. The balloon is inflated, thus angioplastying the lesion, and also allowing for the diffusion of our proprietary molecule into the arterial wall. When that diffusion has been completed, which is very quickly, we then activate the integrated light fiber in the device for 60 seconds, thereby creating the links between the collagen and elastin that I previously described. The best part is then we deflate the device and we remove it, and you leave nothing behind. There's no metal, nothing to stay in the vessel other than the native proteins that were already there. The drug is rapidly metabolized and eliminated. Let me show you some pretty pictures now of the effect of the therapy, most importantly. As you probably all know, pigs, which are the typical model for vascular therapies, don't typically get atherosclerosis. So instead we've utilized an overstretch model where we stretch the vessel about 30 or 40% beyond its normal diameter. If you do that to a normal vessel with a plain balloon, what you see is on the left, the vessel snaps back to its typical diameter. If, however, you treat it with NVS, hopefully you can make this out in this light, you see what appears to be the shape of an angioplasty balloon sitting in the vessel. There is no balloon there. The balloon's been removed, but we've effectively remodeled that vessel around the balloon shape. Perhaps a more dramatic picture here. One of our medical advisors, Krishna Rocha Singh, was in the animal lab with us one day, and we asked him, Krishna, can you take a wire and make a dissection into a vessel, because we think we can repair it? He said, okay, sure. Well, he got a little overly exuberant with his wire, and what he actually did was to perforate the vessel. You can see here the contrast leaking out of the vessel, and you can appreciate the fact that the vessel is quite spasmed down compared to what it normally looks like. In any event, we went ahead and treated it anyway, and you can see here what caused everybody's eyes to widen. Not only have we sealed the perforation in the vessel, but we've maintained that balloon shape despite all the spasm going on in the native vessel there. This is acute results. We then also decided, okay, we need to show how long these results last. So we decided to do a chronic study in animals. Now, I just told you that pigs don't typically get atherosclerosis, but there is actually a model, a genetically modified model, where you can knock out the LDL cholesterol receptor in a pig, and you can put it on a high-fat diet for 9 to 12 months. That actually creates some atherosclerosis in the pig. It's not quite the human disease, but it is disease. And if you stick a metal stent in that model, what you'll see is that the pig gets restenosis. So, this is from a slide at day 42 of that study, 28 days after the administration of either a metal stent, a plain balloon, or NVS therapy to the pig. And what you can appreciate, these are OCT images of the vessel. This is the stent. You can notice the struts. And you can notice that there's thickening of that wall. There's definitely restenosis going on here, whereas in the NVS vessel, nice and wide open, no evidence of restenosis. So we found this to be very compelling and good indication that this should work in a human patient. In order to execute on a program like this, which combines both a drug and a device, you need to have a team that's skilled in both. And I'm quite proud of the team we've managed to assemble. We have what I call the usual suspects that you see in device companies, people like Steve Tyler and Hank Hauser. Steve has spent 25 years leading R&D teams in cardiovascular device companies. Hank started his clinical career in the cath lab and has been doing clinical development since then on devices, combined with folks like Catalin and Kevin, who are from the pharmaceutical industry. Catalin being a PhD vascular biologist who's been doing cardiovascular pharmacology her whole career, and Kevin, who's a pharmaceutical chemist who came from Allergan doing small molecule development. And this team, I think, is uniquely well positioned to advance this to the market. We have also been lucky enough to attract some of the leading lights in the space. If you're familiar at all with the vascular field, you'll know some of the names in this. Alois Edelman is one of the godfathers of drug delivery to the vasculature. Bill Sessa runs the Yale Basic Vascular Biology Lab. And Gary, Bill, and Craig on the right there are constantly on the podium at vascular meetings presenting their findings. Bill, in fact, just did it at Link in Leipzig, and Craig is going to be a clinical site for us. Today, we're at a state where we've performed extensive preclinical proof of concept. We've actually treated well over 70 large animals with our therapy. We've also done a bunch of experimentation in perfused cadaver work, human cadaver work, because we needed to show that we could actually deliver the therapy in the setting of a human disease. Lastly, we've done quite a lot of basic science. I haven't shown you any of it here because I only have 15 minutes, but if you're interested, I'm happy to show it to you, that support the use of NVS therapy in patients. We will have the first patient in our U.S. phase 1 trial this quarter. We have approval from the FDA to do so. And the next step following that 15-patient trial is going to be to move into a 75 to 100 patient efficacy study with our integrated commercial product. And that's in fact what we're out raising money to do. We're planning to raise approximately $30 million US. That would get us through the end of 2022 and would achieve the goal of effectively getting what I like to term phase 2 results, meaning a robust safety and efficacy trial using our commercial product, the fully integrated product, and being able to position us well for a pivotal study for approval. Our existing investor, which is a hospital system in the US, Avera Health, has been supporting us to date, will contribute fresh capital to this investment, but we're now looking for external investors to join the mix as well. Why you should care as an investor about peripheral artery disease companies is that over the last dozen years, due to the unmet need in the space, there have been many companies that have developed novel technologies. This is a list of a few of them that have been quite tremendous investment successes for the folks in that space, and that's one reason as an investor it's worth paying attention. Our investment thesis is quite simple. Number one, companies are out there putting stents and balloons and outdated technology into their sales bags. They're poorly differentiated products from one another, and they have major clinical limitations. And now there's new safety questions around the use of paclitaxel. Nevertheless, they're major profitability drivers for the incumbent companies in the space. And we are coming along with something that has the potential to dramatically improve that therapy and also be quite differentiated from it. Thank you for your attention.